Our next speaker is often referred to by his colleagues as a neuroscience genius. Ed Boyden won the mathematics category at the Texas State Science Contest, at age 12. At age 16, he entered MIT after skipping 2 grades. He graduated at 19. Boy, his career trajectory is just like mine, I'm telling you, it's just exactly it. At 19, he graduated with 1 master's degree and 2 Bachelor of Science degrees, one in physics, the other in computer science. He received his PhD in neuroscience in 6 years, became a tenured professor at MIT in 7, and today he directs 3 different programs at MIT. Ed Boyden has developed 4 different technologies that could someday win a Nobel Prize to go along with the many other prizes that he has won already. He understands the brain and how it functions as few people in the world do. Ladies and gentlemen, Ed Boyden.
Growing up just north of Dallas, Texas, I was really mystified by just existence as a child. You know, why do we do what we do, feel what we feel, think what we think, and why can we be cruel to each other? Why do we make bad decisions? You know, why is humanity the way it is? Can we become better? And over the years, I explored different parts of engineering, of science, and honed in on this idea that maybe we should really try to build technology to help us understand ourselves through understanding the brain. Our brains in our heads are incredibly complicated objects. So in a cubic millimeter of our brain could be 100,000 tiny brain cells called neurons, and they're wired up with these elaborate tree-like patterns that allow them to connect up, like a computer circuit, but far more intricate and elaborate than one can possibly imagine. The brain cells in our heads fire off little electrical pulses, and they communicate. Right now, as I say these words, auditory parts of your brain, visual parts of your brain, they're all registering millisecond timescale electrical pulses. How can we understand this and how it generates our dreams and our thoughts and our feelings? But there's another reason why this quest is important beyond mere curiosity, beyond the hope that we could understand our philosophy, our existence, and our beings. And that is brain diseases, disorders, and conditions that affect people all over the world. If you look at the list, Alzheimer's disease, spinal cord injury, stroke, these diseases do not have a cure, and the treatments are quite partial and have a lot of side effects. These diseases can affect us not only in terms of our time to live, but they can change who we are. So how can we tackle this problem? Well, the complexity of the brain is very daunting. It's this densely packed machine. And so the prospects so far have been slow and difficult. These are some numbers from an editorial in Science magazine from a bit over a decade ago, and I think that the numbers have in some ways gotten even worse. But a clinical trial for a brain drug might take you a decade. The failure rate will be distressingly close to 100%. The cost will be enormous. I think it's now several billion each. And even if you make it through those hurdles, even if your therapy is approved, there's a good chance that it only works partly and that it has side effects for a significant fraction of the people. So what can you do about it? Well, the history of science shows us that when a scientific field hits the ground truth, the fundamental understanding, the list of those building blocks and how they work together, for example, then you can make progress in an accelerated way. Let's look at physics, for example. Quantum mechanics a century ago allowed us to understand the particles, and how they work together, forces. And in the century since, we had the transistor and the laser and the microchip and the internet and so forth. An exponentially growing body of scientific enterprise that had huge impact on everyday life. What's the ground truth for the brain? Well, the brain is extremely complicated. Let's zoom in a little bit. So the spatial scale of the brain is challenging, right? The brain is an enormous object, and yet the wiring of the brain is nanoscale, We're talking about much smaller than the diameter of a human hair. So how do you cross those scales from the tiny to the large? That's a challenge for science that demands new technology. And then there's temporal scales. So over the time scale of aging, or the development of a disease like Alzheimer's, or the learning of a language. These are slow processes that can take years, but the briefest elements in the brain are these millisecond timescale electrical pulses within brain cells and the chemical exchanges that happen within them. So I lead a group at MIT where we build tools to try to help brain scientists conquer the spatial and temporal dimensions. To understand the brain, ideally, we would be able to control those high-speed electrical signals. We can understand what they do and maybe repair brain states that need help. We should watch what's going on in the brain so we can understand what information is there. And ideally, we would map out the wiring of the brain so we know how it's architected, how the structure is organized. Now, that's a lot of data. How can we understand it? I would like to argue that one of the best ways of integrating all that data would be to make a computer simulation of the brain. We could integrate these tools, which I'll talk about in a second, control, recording, and mapping, and derive data sets that could allow us to recapitulate the brain in software. Why would this be useful? Well, as I mentioned just now, one issue, of course, is just the sheer complexity of all that we would have to unveil. But a computer simulation could be used to try to pinpoint where in the brain to optimally intervene. Causality is hard, but if you can screen in software or in a computer, you could go at high throughput. Maybe we would be able to watch what happens as a thought takes shape or an emotion happens and see maybe even someday what a subjective experience might look like. And of course, AI is all the rage right now, but I think it's very clear that making human-like AI could be very important if we want to recapitulate things like human-like ethics and judgment. And one way to achieve that might be to study the brain at a level of detail that would let you recapitulate it. So today I will tell you a couple short stories about these tools and how they work and what we can do with them. Let's start with control. Brain cells use electrical pulses to compute. How do we control them? Well, suppose you could put little solar panels in them and then shine light on them. Light gets converted to electricity, and you could then aim the light wherever you want. And the brain doesn't feel pain, so you could bring optical fibers into the brain the same way that electrodes are brought into the brains of awake human patients for deep brain stimulation. Now, how do we make brain cells have little solar panels? Well, it turns out all over the tree of life, you can find molecules that convert light into electrical energy or electrical signals. Here we have a single-celled microbe, a green algae, and it's swimming around in a body of water. And the way that it knows where the sun is that it has an eye spot, which we just zoomed into. And that eye spot is chock full of proteins that convert light into electrical signals. When they're hit by light from the sun, they open up a little pore and let charged particles across. That's exactly what you'd want, right? If you wanted to make electrical signals in response to light. What happened next was a bit of serendipity. We decided to take the gene that encodes for this protein and put it into the brain. This might have failed, right? Maybe the gene product would have been toxic, or maybe it would have been too slow or ineffective. But serendipity is a big driver of science, and curiosity feeds that serendipity. And we were very, very lucky. We took the gene that encoded for this light-activated protein and put it into the brain using a gene therapy vector. And now the brain cell could manufacture this gene product or protein. And miraculously, it was able to install that protein on the boundary or membrane of that neuron, which is where the electrical activities are generated. And even more serendipitously, perhaps, it worked. We could shine light from a laser or an LED onto this brain cell, and it would then generate electrical pulses, not unlike the ones right now happening in your brains as you hear me say these words. Now, this next movie has some flashing images, so if somebody's photosensitive, maybe you don't watch this until I get back to you. But I want to illustrate how the technology works. So brains, of course, have many kinds of cells, and we can use tricks of gene therapy to put the light-activated protein into some cells and not others. Then, the gene therapy vector will deliver the gene, the brain cells, the neurons will manufacture the proteins and install them, and those cells will become light sensitive and not their neighbors. we can then aim light from an LED or a laser at the brain, and the cells that we have transduced will respond to light, but their neighbors will not. That's how we can control the brain very precisely. Okay, the flashing images are over, if that's of importance. Okay, let's give you examples of how this works in action. I'll start by talking about dopamine neurons. And in the popular press, people often talk about them as the pleasure center of the brain, but of course, brain science is very complex. So in this study, investigators took our light-activated protein that I just mentioned and used a gene therapy vector to make the brain cells of mice that have dopamine respond to light. What does activating these cells do? Well, if it drives reward, you might imagine that if a mouse were to go poke its nose into a sensor and get a pulse of light, it would want to go to that place more often. And if there was a control spot where nothing happened, maybe it would avoid that spot. And that's what happened. Here's a mouse that pokes its nose into a little sensor and gets a pulse of blue light. You can see it going down the optical fiber and does it again and again and again. I think you see the pattern. This mouse is basically working for light. So you can use this technology to prove that a certain brain cell or set of cells does something in a causal sense. But you can also use it to discover therapeutic ideas as well. My colleague Li Wei Sai at MIT used this technology that we call optogenetics, opto for light and genetics because we use genetically encoded reagents, and found that if you take an Alzheimer's model of mice, these are mice engineered to get Alzheimer's-like symptoms, and you pulse the brain using optogenetics at a certain frequency, about 40 times a second, that the molecular pathology can get cleared up. Blood flow increases, inflammation changes, and the Alzheimer's-like problems go down. The teams went on to try to figure out a non-invasive way of doing this, and basically having the mouse watch a movie, flickering lights and hearing clicking sounds was able to recapitulate this. No need for the gene therapy or the implanted optical fiber anymore. And now we have several clinical trials ongoing, including at a startup, trying to do a large-scale clinical trial, try to see if we can use movies to try to stop Alzheimer's disease. Now, what about using the technology in people directly? There are lots of people who are blind because they've lost the light-sensing cells in their eyes. This movie is showing a mouse swimming through a water maze because mice like to swim, but they also like to sit and do nothing. And this is a mouse that has a mutation that recapitulates one of the mutations that can cause photoreceptor loss in people. And so there's a little platform that allows the mouse to crawl out and stay on this dry platform. But being a blind mouse, it takes some time for it to find it. It's not using vision to do this. Now here's a mouse that was blind a few weeks before and got one dose of a gene therapy vector with a light-activated protein encoded, and now it can go right to the target. This was a sort of basic science curiosity for a while, but a couple summers ago, a brave collaborative in Europe took one of our molecules and was able to put it into the eye of a person who was unable to see and was able to do a partial restoration of functional vision. Not perfect, but he could recognize household objects, see doors on a hallway, lines of a crosswalk. There's a long way to go, but it's interesting to think that this technique of optogenetics might not only unveil new patterns of activity that could be of use in restoring human function, but might be useful as a prosthetic in humans directly. That's control. What about recording? Well, you can think of recording as sort of the opposite of optogenetics. We need to flip the sign and make things run the other way. Rather than using light to control the brain, can we have the brain broadcast light or glow or fluoresce? And so to make a long story short, the tree of life gave us those optogenetic molecules that I mentioned, but to record brain activity, we haven't been quite as lucky. And so we had to do in the lab what the world would not do for us. We built robots that can do evolution and that can screen through mutations of a gene to find mutants that we like and that have the properties we desire. In this case, we looked for molecules that would glow when a neuron was active. And many other people have been looking for such techniques as well. We can then build a very fast microscope. This is sort of a schematic of the optics of a very, very fast microscope that can image brain activity. And starting with small brains like worms and fish, but our hope is to go up to mice and who knows, maybe someday humans, you can start to image the electrical traces of neurons across an entire brain, all the way from sensation to movement and everything in between. What about mapping? Brain cells, again, fill our head. They're large, and yet they're tiny in their building blocks. how do we understand such a thing that crosses from the nano to the macro? Can we understand how brain cells form networks and how the molecules are organized within and between those brain cells? I think we've all seen images like this, brain scans. They're non-invasive and they work, for example, in normal human volunteers, but they have a limited spatial resolution. So each of these blobs that you see here, or voxels, might contain millions of brain cells. Nevertheless, many pioneering discoveries have been made using such technologies. At the other extreme, we have microscopes, and those can see smaller things, but light, which it uses, has a finite size or wavelength, and you can't see nanoscale things very easily. So in our group, we practice a lot of learnable, teachable creativity skills, and one of my favorites is to try to be as opposite of what people have been doing as possible. And so we started taking inspiration from another completely different field, chemistry. Now, in a baby diaper, you have swallowable materials. And if you add water, as shown in this cartoon, to a swallowable material, sodium polyacrylate for the aficionados, the water gets drawn in through osmosis, and you get this huge swelling, an experiment that millions of babies do every day. And so we started wondering, for 300 years, the way people image biology things, like cells, is you magnify some kind of image with a microscope, right? Could we magnify an object itself? So the idea became what we call expansion microscopy. Can we chemically weave a dense spider web-like mesh of that baby diaper polymer inside the brain, inside brain cells, in between those biomolecules, the nanoscale building blocks of life? And so, of course, this won't work on a living brain, but you could use preserved brains from brain banks, for example, to do this. So I'll show you that it works, and then I'll tell you how it works. But here in panel B is a piece of the mouse brain, and in panel C, we've blown it up 100 times in volume. And this baby diaper polymer starts out very dense, like in the upper left, and it ends up swollen, like in the lower left. Here's a little time-lapse movie of probably about half an hour, split up to half a minute. And this is a piece of brain where we formed the polymer earlier, and we add the water right there. I think you can see this piece of brain tissue is growing before your very eyes. All right, so our goal, of course, was to make this so that we could expand everything down to the smallest building blocks of life. And so, as shown in this cartoon, our hope was that we could take a cell like this golden neuron on the left and make it look like the thing on the right, a constellation of biomolecules hovering in space. If two molecules were touching, now there's some minimum distance apart. If two molecules are some bigger distance apart, we scale up by a linear factor. It should look like you draw a picture on a balloon and you blow it up. It should be the same picture, right? Just bigger. Now, how does it work? In the following couple cartoons, we've zoomed in into a tiny patch of a cell just to envision what it might look like. There are 4 steps of what we call expansion microscopy. We, first of all, take molecules like these proteins shown in brown, and there's all sorts of other biomolecules, and we attach little handles to them. That's how we can pull them apart. So the handles are shown in purple. Next, we have to weave that dense spiderweb-like mesh of polymer, and we use a chemical strategy called polymerization. So we can add in so-called monomers, the building blocks of the polymer, and they self-assemble into these long chains that will wind their way inside cells and outside cells, in between the biomolecules and around the biomolecules. And when one of those polymer chains encounters a handle, it forms a molecular bond. We're almost there, right? But there's one more trick that we had to develop, a subtle one. The molecules are very happy where they are, right? They're all stuck together in whatever configuration they normally are. And so we have to soften things up. We can use detergent, heat, enzymes that can loosen things up. And then we add water, and because of the softening, and because of that polymer, and because of the handles, we can pull all the building blocks of life apart from each other. So again, this does not end in a living state. This only applies to preserved specimens, but still, the information we care about, the nanoconfiguration of life, is what we get to see. Finally, we have to label things so we can see them. We can bring in tags that glow, and then we can image them under a regular old microscope and see the structure of the brain. So here's some actual brain data. This is from a region of the brain of a mouse that's involved with memory formation, amongst other important functions. and the cells are expressing glowing proteins from corals and jellyfish and other natural organisms. And we expanded it. And I hope you can see, we're about to zoom into this piece of expanded brain tissue, that the fine wiring of the brain can be seen. Here's a fruit fly brain. It's a very small brain, but again, there's a lot of, we would learn by solving a very small brain completely, and so we're talking to lots of people about how that might look. This is a fruit fly brain where the dopamine neurons are expressing a fluorescent protein, and we just zoomed in from the outside, and now we're going to go out to the lateral edge, and then we'll zoom back out again. So you can see how we can capture, in a very simple experiment, the scales across which the brain is organized. By the way, we share all of our tools freely. The optogenetic tools I mentioned earlier are being used by thousands and thousands of scientists, and the same is true for expansion microscopy. I really believe in democratization of science. And so just as a side note, for example, people are using expansion to try to do things like early disease diagnosis. Many of the leading causes of death, if you can detect them earlier, you can save more lives. So for example, early in a cancer, as shown in this photomicrograph, if you could expand a biopsy and make the small early changes more visible, maybe you could save more lives. And so we're working with many people, pathologists, infectious disease experts, and so forth, to try to see if we can help them solve problems in biology and medicine. We teach this technology of expansion very broadly, and recently the 10th anniversary of our first announcement happened a few months ago, and one of my favorite quotes was from a former group member who's now running a group at Carnegie Mellon who said, "It's so easy, the expansion technology, even my 9-year-old can do it." Okay, so we share all the tools freely, but how do we put them together? As I mentioned, we would love to be able to control brain circuits, image their activity, and then make a map of how that all looks in terms of the anatomy, and combine those data sets into computer models. And so we're starting to make such a plan. It's very early days, but this diagram kind of shows what we're hoping to do. In blue, can we perturb the brain and collect neural activity data? And in yellow, make a map of how it's organized. Maybe starting with small brains like fish and worms, but scaling up to mouse and human as fast as possible. And then we can make different kinds of model. Deep learning and machine learning can help us infer what the network is doing. We could try to infer also what the physiology that underlies the mechanisms of emergent dynamics looks like. And then, of course, we have the ground truth maps of the molecules, and we can connect these data sets. The hope is to start small and then have a bit of a Moore's law-like effect. So a small worm has only a couple hundred neurons. A larval fish has about 1,000 times more than that. A mouse is 1,000 times bigger than that. And a human is about 1,000 times bigger than the mouse. So by working small and starting to scale up to big, we hope to understand the brain, but also to prepare the technology for scale. It's early days. People like to talk about interdisciplinary science, but I hope to end by just conveying to you that neurotechnology and brain science have become omnidisciplinary. Optics and genetics, polymer chemistry and computer science, we're seeing every part of the sciences team up to tackle the brain. And so this last slide is too long to go into, but I just want to acknowledge the people who have contributed to these projects over the years. It's a truly global and omnidisciplinary effort, and I hope that we can bring a lot of insight into the human condition in the time to come as a community, and hopefully alleviate some of the suffering that I guess will affect all of us if we live long enough that affects so many of us currently. Thank you.